In a natural population, there will always be genetic variations. However, in the case of viruses, variations occur mainly due to errors in genetic replication. When viral infection occurs in host cells, the invading virus reproduces by hijacking the host cell mechanisms to replicate its viral genome. The replicated genome will then be translated into a chain of amino acids, which is used to build viral proteins, by folding into a specific shape that packages copies of the genetic sequence, and forming a new virus. During viral replication, some errors may occur. Sometimes, these mutations result in synonymous mutations, in which no change in amino acids occurs. Sometimes, these mutations result in non-synonymous mutations, in which changes in amino acids occur, such as a deletion of an amino acid in the chain, a duplication of an amino acid in the chain, or a substitution of one amino acid for another amino acid in the chain. This may lead to alteration in the features of the final protein product. These mutations can be given names such as N501Y, which means that the amino acid asparagine, N, at position 501 in the chain has been substituted by amino acid tyrosine, Y. Or del247, which means that the 247th amino acid has been deleted in the chain. Some mutations may be harmful to viruses, in which it decreases the survival advantage of the virus, causing the mutation to slowly be eliminated from the population. However, some mutations may be beneficial to viruses, in which it increases the survival advantage of the virus, through mechanisms such as easier viral entry into host cells or host immune evasion, causing the mutation to remain and dominate in the population. When mutations accumulate in a population of a virus, making them differ enough from the original population, it is defined as a new strain. New strains of viruses appear when the virus is allowed to spread rapidly in a community or when the virus infects hosts with weakened immune systems. In the case of SARS-CoV-2, many variants have been identified through genetic sequencing. These variants of virus can be classified into variants under monitoring, variants of interest or variants under investigation, variants of concern and variants of high consequence. Variants under monitoring are variants with mutations suspected to cause a change in transmissibility, virulence, or diagnostic, therapeutic, vaccine efficacy. Variants of interest are variants with mutations suspected or confirmed to cause a change in transmissibility, virulence, or diagnostic, therapeutic, vaccine efficacy, plus community transmission, a cluster of cases, or detection in multiple countries. Variants of concern are variants with increased transmissibility, virulence, and/or decreased diagnostic, therapeutic, or vaccine efficacy. And variants of high consequence are variants that demonstrated failure of diagnostics, with evidence to suggest a significantly reduction in vaccine effectiveness, a disproportionately high number of vaccine breakthrough cases, or very low vaccine-induced protection against severe disease, significantly reduced susceptibility to multiple emergency use authorization, EUA, or approved therapeutics, more severe clinical disease and increased hospitalizations. No variants have been classified as variants of high consequence so far. As of July 2021, 12 variants have been classified as variants of interest, including the Epsilon variant, Eta variant, Iota variant, Kappa variant, Lambda variant, Zeta variant and Theta variant. From the variants of interest, a few variants have gained public attention, such as the Lambda variant and Epsilon variant. Whether it will be reclassified into variants of concern remains to be monitored. Those classified as variants of concern include the four main variants, which are the Alpha variant, Beta variant, Gamma variant and Delta variant. These variants share some common mutations which increases the potential for viral transmission, causing these variants to easily dominate in the population, and resulting in new waves of outbreaks in many countries. Some variants have also shown potential to evade hosts' immune response, including antibodies from natural infection or vaccination. The vaccines available currently are still effective in lowering the rate of severe disease, including hospitalizations and deaths due to COVID-19, while some may have reduced efficacies in preventing viral infection. More research is being carried out on the effects of these mutations on the efficacies of various types of vaccines. Therefore, it is vital for the spreading of SARS-CoV-2 to be contained, as when the rate of viral replication is reduced, so does the rate of mutation. Remember to always wear your mask in public, avoid large gatherings and stay at home whenever possible, even after being vaccinated. Only we can break the SARS-CoV-2 chain of transmission. If you would like to know more about each variant of concern, stay tuned to this series on my channel. If you enjoyed the video, don't forget to like, subscribe or share.